Hello students. So today we are going to learn part 3 of chapter 6 plant water relation. This is the third and last part of the chapter in which we are going to learn the topics transport of mineral ions, transport of food, transpiration. There are three types: cuticular transpiration, lenticular transpiration, stomatal transpiration. Also we will be learning structure of stomatal apparatus, opening and closing of stoma, significance of transpiration, and finally the last part, that is transpiration is a necessary evil. Now moving further, the transport of mineral ions. Here first you can see the animation slide towards the right, where you can see the movement of water molecules in the xylem, where xylem vessel, xylem fibers, structure, xylem parenchyma is present, very clearly labeled. So you can see step by step vessels, how the water is moving, xylem fibers, structure, xylem parenchyma, and you can see the movement inside the plant. So first point, soil serves as a main source for minerals. Minerals constitute most commonly occurring solid, crystalline inorganic materials obtained from the earth's crust. So here as we can see, the minerals which is obtained from the soil, so which is the main source, and it is usually in the form of solid and crystalline inorganic components. Minerals play an important role in the day-to-day life of plant. Minerals are absorbed by plants in the ionic, that is dissolved form, mainly through roots and then transported. So the minerals are important in the daily life of plant, as in case of any living organism. So without the minerals, the growth may be affected. All the minerals may be in minute quantity, large amount, different proportions they are required, but they are essential for the growth of different parts of the plant, and it is absorbed in the dissolved from the roots, and it transported to the various parts of the plant. Moving further, absorption of minerals is independent of that of water. Absorbed minerals ions are pulled in upward direction along with xylem sap because of transpirational pull. So this absorption of minerals does not depend on water molecule, but they are pulled along with the water in the upward direction through xylem sap, which because of the transpirational pull. Now, moving further, mineral ions are needed in the areas of the plant, that is apical, lateral, young leaves, developing flowers, fruits, seeds, and storage organs. That means each and every part of the plant requires minerals. As various types of minerals are present, all are required in certain proportion. Right from the top of the plant or the lateral region, the growing new young leaves or the flowers, fruits, seeds, storage organs, each and every part requires minerals. So soil is not the only source for mineral uptake. Also from older parts export their ions to the younger leaf before the fall. Most readily mobilized ions are like phosphorus, sulfur, nitrogen, and potassium. Now, as we just learned, soil is a major source of mineral. But then, other than that, even the older parts, they also transport the ions to the younger leaf, and that is before they fall. These are passed on to the younger leaves, and the most important and the readily available ones are phosphorus, sulfur, nitrogen, and potassium. Further, analysis of xylem exudate also shows that some nitrogen travels as inorganic ions, whereas some of it is carried in the form of amino acids and related compounds, that is SNP, which is sulfur and phosphorus. So along with the xylem component, the nitrogen also travels or is present along with the inorganic, or you can say the organic form, like amino acids, which also have are present in different components like sulfur and phosphor. Some exchange of materials also occur between xylem and phloem. So there is a exchange of material taking place in xylem and phloem. It's not that xylem continuously carries water and phloem food, but even there's exchange between them. That's why the present animation which you can see very clearly, we can see how the component not only travels ascending or descending. As we know, ascending increasing order, that is in case of xylem, and descending in case of the phloem. But here you can see transversely also the molecules being passed from the cortex region, endodermis, phloem to cambium and xylem component. So you can see very clearly how lateral movement of molecules takes place, which clearly indicates that there is exchange between xylem and phloem. Now, further we come to the next part, that is transport of food. Now we learned about the transport of water, which is mainly done by the xylem. So with the food, that is in case of phloem. Now first see the labeling part: sieve tube, companion cells, phloem fibers, phloem parenchyma, sieve elements, which are the parts of phloem. So very clearly we can see in this animation slide various parts of the phloem. Now first point, all the plant parts require continuous supply of food for nutrition and development. That is, as we know, the growth and development of any living organism requires nutrition, right? So similarly, plants also requires continuous nutrition of and development is required. So in higher plants, there's a great differentiation and division of labor. So it is differentiated and the division of labor is also present. Chloroplasts are confined to green cells of leaf where food is synthesized. Now this we are learning from right from school time in the lower class, that photosynthesis which is carried out in the leaf, that is in the chloroplast, in the presence of sunlight, in also the source which is utilized or the raw material is water and carbon dioxide. So it is mainly confined for production of food. Further, the non-green parts like root and stem must receive food from leaves. So as food is prepared in the leaf, so from the leaf, it has to be passed to the non-green parts, that is mainly the stem and the root. The part where food is synthesized is called source. Students, see the two terms we are coming up. One is the source where the production is done, while the part where is utilized is the sink. So we come across two terms, source and sink. Food has to travel from source to sink region. This movement of food from one part to other part is called translocation of food. Okay, I hope that part is clear. Coming to the minerals which are needed by the plants. Minerals that play role in the day-to-day life are called essential elements. About 36 to 40 elements are incorporated in plants' life. Some minerals like C, H, O, P, N, S, Mg required in large quantity are macro elements. Now I hope these terms or the elements we come across in the chemistry part, right? That is C, carbon, H, hydrogen, O, oxygen, P, phosphorus, N, nitrogen, S, sulfur, Mg, magnesium, whereas they are the macro element. The minerals like Cu, that is copper, Co, cobalt, Mn, manganese, B, boron, and Zn, zinc, required in small quantity, which are micro elements. So the minerals are required in large quantity, that is the macro elements, and smaller amount or in minute trace quantity, which is the micro elements. So again you can see a image which I put over here, which shows the water minerals which is taken up by the root. You can see the arrow moving upward direction along with the water, that is through the xylem components, which reaches from the stem till the top part, that is the leaf, and from the leaf by transpiration it is removed. Whereas carbon dioxide which is taken in, oxygen is given out again through the stomata. Sugar, or we can see the glucose which is synthesized in the leaf. From the leaf it is passed to the various part of the plant body, that is stem and the roots. So you can see xylem usually taking upward direction, whereas the phloem carrying the food from top to bottom, ascending and descending. Now, path of translocation. As we just read the term translocation, that is carrying the food to different parts. So here we learn how this is translocated. Food is to be translocated to longer distance is in higher plants. Hence, plants must have adequate channels for transport of food. Sieve tubes and vessels are structurally ideally suited for vertical translocation. Now, as food has to be passed to various parts of the plant, that is stem and leaves, so these are the proper channel is required, or for transportation proper route is required. So this is particularly suited by the presence of sieve tubes and vessels, which by vertical translocation carries the food or helps in translocation. Further, the ringing experiment structure and distribution of phloem, chemical analysis of phloem sap, and use of isotope, that is carbon 14, clearly points out that phloem tissue is primarily responsible for flow of food in the vertically downward direction. So the experimentation was carried out using isotope carbon 14 called as the ringing experiment, which showed that food which is prepared from the leaf, that is by photosynthesis process, and it is carried from upward to the downward direction vertically through the phloem component, just as we saw in the previous slide, the sieve tubes, vessels, all other components of phloem, which helps in translocation. The horizontal translocation occurs from phloem to pith or phloem to cortex via medullary rays in the stem. Even along with the vertical flow, horizontal translocation also occurs from phloem to pith or phloem to cortex region. Food is always translocated in the form of sucrose and always along concentration gradient from source to sink. The transport of food occurs in vertical and lateral direction. That is, as we know, the glucose is prepared in the food. It is transported or translocated in the form of sucrose, and we learned two terms earlier, source and sink. So it is carried from source to sink. That is, from the place where it is produced and to the place where it is required, that is stem and root. Also, along with the vertical transportation from top to bottom, from the leaf to the various parts, also it moves in the lateral direction as well, which again you can see in this particular animation which I put up: xylem, phloem, pericycle, caspian strip, the endoderm. You can see how the molecule moves in the cortex epidermis region. So you can see how the movement taking place over here vertically downwards, and also to some extent in the lateral way. So you can see the lateral movement as well as the uh, vertically downward movement of the foot which is translocated. So again, a very good animation to explain the concept. Now, vertical translocation. Now, again, first you can see the image over here, how the translocation is taking place. Now, this one which is moving upward, as we know, it is the xylem, correct, which carries the water along with the minerals. Now, see the right side one, the phloem vessel, where you can see the movement not only upward, also the downward vertical movement also taking place, and slightly partial horizontal movement. So here in vertical transport, food is translocated in the downward direction from leaves to stem and root. It also occurs in the upward direction during germination of seeds, bulbils, corm, etc. So now here onto the second part of the animation, you can see movement down and up. Why it is upward? Just now as we learned, it is in the downward direction, descending way, right? But here it is seen the upward. Reason? Because as the flowers or the birds, the fruits or other modified region of the plant which also require energy, the nutrient which is supplied in the upward direction to the lateral region or in the various parts where the growth occurs, as given the example seeds, bulbils, corm, etc. So the upward translocation also occurs from leaves to growing point of stem to developing flowers and fruits situated near the ends of the branches of the stem where it is required. Further, the lateral translocation. It occurs in the root and stem where the lateral translocation, the root and the stem region. When food is translocated from phloem to pit, it is called radial translocation. See here the terms again, students. Phloem to pit, if the translocation of food occurs, it is radial translocation. And from phloem to cortex, it is tangential translocation. Here you can just relate to the mathematics part, the term radial, the term tangent or tangential translocation. The transport of food through phloem is bidirectional, as you can see in this particular image, as it is going upward as well as downward direction. So it moves either way. Phloem sap contains mainly water and food in the form of sucrose, as we learned earlier as well, but sugars, amino acid and hormones are also transported through the phloem. So along with the food, which is the major thing which is transported from the phloem, but along with that, certain sugars, amino acids, hormones are also transported through the phloem to the various parts of the plant. Now, mechanism of sugar transport through phloem. So we come across how this sugar is transported. So the first point, several theories like diffusion, activated diffusion, protoplasmic streaming, electroosmosis, pressure flow, etc. are put forth. The most convincing theory is Munch pressure flow theory or mass flow hypothesis. Now, the certain theories which you already learned earlier, that is a diffusion, activated diffusion, protoplasmic streaming, pressure flow, electroosmosis, etc. But the one which is explained clearly about the flow, it is the Munch pressure flow theory. That is, Ernest Munch proposed that photosynthetic cell synthesizes glucose, hence its osmotic concentration increases due to endosmosis from the surrounding cells, and the xylem is absorbed. So as we know, endosmosis, that is moving from outside to inside part. The cell becomes turgid, means it swells up. Due to increase in turgor pressure, sugar from photosynthetic cell is forced ultimately into the sieve tube of the vein. This is called loading of vein. Now the term loading, we can just relate to our daily life, that is loading and unloading with certain things which is loaded into a vehicle, where what is required, where it is has to be sent. So here also can relate with the term loading of the vein, where the sugar is loaded from the photosynthetic region of cell to the sieve tube. Now here I just added an image which could help you to understand the concept. That is, if you see here, xylem component in blue whereas phloem in pink, how the movement takes place. There is components moving from phloem to xylem region through the water. Here you can see transpirational stream or the flow, transpiration which occurs from bottom towards top region, pressure flow from the top to bottom, enters by osmosis because of high concentration of the sucrose. Now adjacently you have got companion cells, the source, that is source, the leaf where the food is prepared or synthesized in the chloroplast, and from there it is moved into the companion cell, and companion cell into the phloem, and from the phloem it is taken to various part of the plant, that is stem, root, etc. Similarly, you can say it is even passed to the nearby companion cell and the sink. So through the companion cell, it reaches the sink, that is where it is required. Here again you can see on the top part ATP, which is broken down to ADP. Here the H plus ions, the sucrose which is formed. Now, further, at the sink end, root cell utilizes sugar and polymerizes excess sugar into starch. Its osmotic concentration is lowered. Exosmosis occurs. Water in the root cell is lost to the surrounding cells, thereby decreasing the turgidity of the cell. Turgor pressure is lowered. So, the sink end, that is the place where the food has to be translocated, where the sugar is utilized, or the sugar is again converted into starch if there is an excess amount present. So it takes place by the exosmosis. Further, that because of which exosmosis, the turgidity of the cell also decreases, lowers. So pressure is lowered. Hence, a turgor pressure gradient is developed from the sieve tube into the leaf to the root cell. Consequently, food is translocated on the concentrated gradient. Passively, this is vein unloading. At the sink end, sugar is used and excess water exudes into the xylem. So again here, vein loading takes place initially, as you saw in the earlier image. After loading, it passes through the companion cell into the other components of phloem, the vessel, from which it takes to the again companion cells, and from through the companion cell to the particular region where it has to reach, that is the stem, root, or as you use the term, sink region. So where the vein unloading takes place, or as I gave you example, the loading of things from home or various places, factories, industries, and then again unloading. So here the vein is unloading the things. Main objection to this theory, so there is certain objection also. What is that? That is, mechanism does not explain bidirectional transport of food. Moreover, according to Munch, pressure flow is purely a physical process. So most important, as we saw the previous slide, the animation where the phloem region is bidirectional, that is food is moving upward as well as downward region. Reason, upward region towards the flower, fruit, seeds, etc. Downward towards the stem or the root region, right? But it is not explained. So here, there's again a chart I added for a better understanding, the pressure flow hypothesis. Physiological process of loading sucrose into the sieve elements at the source. Again, it is something related to the earlier image which you saw in the earlier slide. Then here water molecule which is there in the xylem region, this is the phloem region. From the this particular sink region, or you can say sink region, where it reaches through the source, that is over here. How it reaches the sink cell, you can say, that is the sucrose which is reaching at this point. Also there is a flow from the phloem into the xylem component as well. So pressure flow, phloem and xylem are coupled in an osmotic pressure that transport sucrose and circulates water. Physiological process of unloading sucrose from sieve elements into the sink. Here it is the loading process at the top region, here it is the unloading process. Further, again I added another image, mass flow hypothesis Munch, which was put in 1930, again to understand this concept, that is loading, unloading, etc. So step 1, water absorption from nearby cells increasing hydrostatic pressure in mesophyll cells. That is, this is the region where the food is prepared, as we know, by photosynthesis in the chloroplast. From here it is taken to the companion cell, from the companion cells into the sieve tubes, the vessels through which it is passed forward, then into the companion cell adjacent to the sink or the root region, and then into the sink where is the root cell or where the food has to be reached, right? The second step, whereas just as you saw phloem loading taking place, transfer of organic food from mesophyll cells to the sieve tube, that is from here to here. Then the third step, as I mentioned, water entry from xylem to sieve elements by osmosis. Step 4, which you can see here, hydrostatic pressure difference drives solute movement from sieve tube to the sink region where it has to reach. Step 5, phloem unloading, transfer of organic food from sieve tube to sink. Then step 6, water moves back to xylem vessels by osmosis and transpirational pull up water. So this is a very simple image which clearly understands the mass flow hypothesis by Munch, how the loading unloading takes place right from the preparation of food by photosynthesis from the leaf, and how it passes through various components of phloem and reaching the region of stem or the sink, as we use the term, the root cell or the different regions of root. Okay, I hope that part is very clear now. Coming to the next part of the chapter, it is transpiration. Now until now you are seeing the transpiration process taking place to the stomata. So here you can see the image showing the water which is taken up from the soil, moves from the root through the stem, from the stem towards the upper part, that is the leaf region, and from the leaf the water evaporates by transpirational process. So we come to the first point, Plants absorb water constantly and continuously. Hardly 5% of the total water absorbed by roots that is utilized for cell expansion and plant growth. Remaining 95% water becomes surplus, which is then lost into the atmosphere through its aerial parts. See, there's a lot of hard work going on over here. That is, the plants absorb water from the root, but out of that water, or we can say 100%, then 95 percentage of water, it is evaporated by transpiration. Only 5 percentage is utilized. See how much difference, even though whatever water is taken, only 5 percentage is only utilized. Hardly 1% of surplus water is lost in the form of liquid, and 99% of surplus water is lost in the form of vapor. The loss of water in the form of liquid is called guttation. This term again we come across in the earlier beginning slide, guttation process. It occurs through special structure called stomata or hydatodes. Now hydatodes are present at the marginal region of the leaf. This specifically you can see in fern plant, how the guttation process takes place by the hydatodes. The loss of water in the form of vapor is called transpiration. Now this term you have come across many times. Transpiration, loss of water in the form of water vapor. If I add a point, that is the aerial part of the plant, that is leaf, because most of the percentage is removed out or vaporized through the leaf. That occurs through leaves, some through stems, flowers and fruits as well, but a minor percent. Most of the transpiration occurs through the leaves, called foliar transpiration, means through the leaf. The actual water loss during transpiration occurs through three main sites. So there are three main regions through which the transpiration takes place. Mainly, as we learned, there is a leaf region, but three parts are the cuticle, stomata and lenticels. Accordingly, three types of transpiration are recognized. So based on which we are learning three types. One is cuticular, second stomatal and lenticular, which we'll be learning in the next slide. Now, coming to the first type, that is cuticular transpiration. Cuticular is a layer of waxy substance, cutin, present on the outer surface of the epidermal cells of leaf and the stem. So cutin, which is present on the epidermal layer of the leaf. So the transpiration takes place through leaf only. But if you see the further point, cuticular transpiration occurs by simple diffusion and contributes only 8 to 10% of the total transpiration. Unlike as we read, the 95% or 99% is evaporated. So only 8 to 10% by cuticle. Cuticular transpiration occurs throughout the day and its rate is inversely proportional to the thickness of the cuticle. That means what? In simple words, the cuticle thickness is more, water transport will be less. If the cuticular thickness is lesser, transpiration will be slightly more. So here, based on that, I added an image over here, that is cuticular transpiration. The loss of water through the cuticle is known as cuticular transpiration. In simple words, the water which is evaporated through the cuticular region. So you can see the cross section of the leaf, the cuticle which is present at the uppermost part of the leaf. Now, coming to the second type, lenticular transpiration. Lenticels are small raised structures composed of loosely arranged complementary cells. So, they are the additional cells or the complementary cells. Each lenticel is a porous tissue consisting of cells with large intercellular spaces in the periderm of the secondary thickened organs and the bark of the woody stems and roots of dicot flowering plants, the region where all these lenticels are present. I repeat this part: periderm of the secondary thickened layered organs, bark of the woody stems or the root of the dicotinous flowering plants. Further, lenticels are present in bark of old stem and pericarp of woody fruits but are absent in the leaves. So they are not present in the leaves. If you see here, you can see the region where the lenticel is present, and if you see here, there are a lot of space present between. That's what we read, large intercellular spaces in the periderm. Further, lenticular transpiration contributes only about 0.1 to maximum 1% of total transpiration. Means the least amount of transpiration takes place through the lenticels. Rate of lenticular transpiration is very slow. It occurs throughout the day. Clear? Now, coming to the third type and most common type and most percentage of water which is transpired. So here stomata are a minute apertures formed of two guard cells and accessory cells. They are located in the epidermis of young stem and leaves. Leaves generally show more number of stomata on the lower surface. Now if you see here the leaf, in the leaf you can see the magnified image where you can see the stomata, and the stomata, again you can see how the structure which is present and the different components present, the chloroplast which is present, the vacuole, the guard cells, stomatal pore over here, right? So different components and how individual stoma looks like. This particular image when the stoma is open and here when the stomata is closed. Now, second point, depending upon distribution of stomata on leaves, leaves are categorized into three types, namely epistomatic on the upper epidermis, example hydrophytes, example lotus. Now here, to understand this term concept, I added all the examples in the next page. That is to understand the different types like epistomatic, example leaf. Second, hypostomatic on the lower epidermis, that is xerophytes. Now as you know, xerophytes were learned in the practical. Xero means desert, phytes that is plants present in the desert region, example nerium. And amphistomatic, amphi another common term like amphibian species present in land and water, like on the both the surface. So here the term amphistomatic means the stomata present on either side, upper and lower side. So here mesophytes, example grass. Stomatal transpiration occurs only during daytime, exception desert plants where it takes place during night time. So 90 to 93% of total transpiration occurs through stomata, and that too during daytime only. Okay. Now, you can see the examples which I mentioned, that is xerophytic, example nerium, mesophyte, example grass, cypress. This example we'll be learning in the next slide. So I added over here. So altogether we can have a comparison. Then hydrophytes, example lotus. Now, structure of stomatal apparatus. So, typical stomatal apparatus consists of two guard cells, stoma and accessory cells. So, two things are there. One is stoma, other accessory cells. Stomata are minute elliptical pores bound by two kidney or dumbbell-shaped guard cells. Guard cell is a type of epidermal tissue which may be called as modified epidermal parenchyma cells. They are kidney-shaped in dicot, whereas dumbbell shaped in grasses. So here, now come to the image before continuing with the next point. Stoma or the stomata, the epidermal cells on the outer lining, chloroplast present inside, the vacuoles, the guard cells over here, the stomatal pore in the center, nucleus over here, subsidiary cell, that is additional cell, thick inner layer, and the epidermal cell, just as we learned. And dicots, if you see the kidney shaped cell open and close, which is the animation image, so you can see both ways, opening and closing. And monocots where it is dumbbell shape, in a best example being grass. And again here how it opens and closes. Coming to the next point, in cypress, that's what the image which you saw in the earlier slide, both kidney and dumbbell shaped guard cells are present. So it is one of the unique species where both are present, dumbbell as well as kidney shaped cells. So students, one of the CT questions, that is NEET questions. Cypress having both kidney and dumbbell shaped guard cells. Guard cells are living nucleated cells with unevenly thick walls. Inner wall of guard cells is thick and inelastic, and its lateral wall is thin and elastic. So in case of the guard cells, they are thick and inelastic, whereas lateral wall which is thin and elastic. Now, Further, guard cells contain few chloroplasts which are capable of poor photosynthesis. Means the chloroplast is present but very few in number. Guard cells have ability to change their size and form, due to which stoma opens or closes. That's why the shape can change for when it opens and closes. Stoma is an elliptical pore formed due to specific arrangement of guard cell. It is through the stoma excess water is lost in the form of vapor. That is how water vapor is evaporated. Accessory cells. These are specialized epidermal cells surrounding the guard cells. Their number is also variable, means the number varies according to species, and are reservoirs of potassium ions, so which store the K plus ions or the potassium ions, which you can see over here. Now this animation which I added is to understand how the oxygen is released, the potassium ions are taken in. I can see carbon dioxide which is again taken in, water vapor and oxygen which is released. So this simple animation showing giving out oxygen and water vapor, taking off carbon dioxide and potassium ions as the reservoirs. So which are the subsidiary cells. So again, if you see, I added one more image which is a picture, not animation, so showing an open stomata and closed stomata. Open stomata where you can see the guard cells, the stomatal pore, the nucleus, inner thick wall, epidermal cell, chloroplast, outer thin wall, what we saw in the earlier image as well. Similarly, the chloroplast, vacuole, the guard cells over here when stoma is closed. Now, opening and closing of stomata or stoma, as we just saw the images. Opening and closing of stoma is controlled by guard cell, turgor of guard cell. During daytime, guard cells become turgid, means it swells up due to endosmosis. What is endosmosis? Movement of water molecules from outside to inside. Thus, turgor pressure is exerted on the thin walls of guard cells. Being elastic and thin, lateral walls are stretched out. Due to kidney or dumbbell-like shape, inner thick walls are pulled apart to open the stoma. During night time, guard cells become flaccid due to exosmosis. Flaccid means it gets compressed, and exosmosis, that is outside to that is inside to outside, which is the exosmosis, as we learned in the earlier point, endosmosis outside to inside. So flaccidity closes the stomata almost completely. Endosmosis and exosmosis occur due to diurnal changes in osmotic potential of guard cells. Different theories are proposed to explain diurnal changes. So here, as we can see, endosmosis taking during the daytime, exosmosis during the night time. That is, stoma remains open during the daytime, closed during the night time. So these diurnal changes are because of the endosmosis, exosmosis, and the osmotic potential of the guard cells. So even these theories, the various theories are put forward to explain these concepts. According to starch-sugar interconversion theory, one of the theory which explains the concept by Stewart in 1964, during daytime enzyme phosphorylase convert starch to sugar, thus increasing osmotic potential of guard cells, closing entry of water, thereby guard cells are stretched and stoma widens. One of the reasons which is very clearly accepted. The reverse reaction, that is opposite thing, occurs at the night, bringing down the closure of stomata. That is, it becomes flaccid and daytime turgid. So here you can see starch getting converted to sugar by phosphorylase enzyme in the daytime. In the night, sugar again converted into starch. That is, stoma opens in the daytime and closes during the night. Further, according to theory of proton transport by Leavitt in 1974, stomatal movement occurs due to transport of protons, H plus and KH plus ions, which we saw in the previous slide as well, how the potassium ions is a reservoir. During daytime, starch is converted into malic acid. Malic acid dissociate to form malate and protons. Protons are transported to subsidiary cells and potassium ions are imported from them. Potassium malate is formed that increases osmolarity and causes endosmosis, that is opening of the stomata and transpiration taking place in the daytime. Uptake of K plus ions is always accompanied by Cl plus ions, that is chlorine ions. At the night time, uptake of K plus and Cl ions is prevented by abscisic acid, which we learn in the hormones, plant hormones part about the abscisic acid, changing the permeability of the guard cell. Due to this, guard cell become hypotonic and thereby becoming flaccid. So here just added an image showing transpiration process through the stoma, which is open, the small openings on the upper and lower side both leaves, which is the stomata. Here I showed another image, guttation, which also takes place in the leaves with the help of hydatodes. Now, significance of transpiration. Advantage, it removes excess of water. Second, it helps in passive absorption of water and minerals from the soil. Third, it helps in ascent of sap. Fourth, as stomata open, gaseous exchange requires for photosynthesis and respiration is facilitated. It maintains turgor of the cells. Transpiration helps in reducing the temperature of leaf and in imparting cooling effect. So see the advantage to remove excess water for passive absorption of water and mineral, causing ascent of sap, stomata which opens not only for transpiration but the gaseous exchange which is required for photosynthesis to maintain the turgidity, also to impart cooling effect to the plant. Now what is the disadvantage? Excessive transpiration leading to wilting and injury to the plant. As we learned, 95 to 99% of water is transpired. So it is a added pressure onto the plant, so or you can say excessive transpiration, it may also lead to the death of plant. So here added a simple animation showing tree where water which is flowing, through which the water is taken up from the root, it passes the stem, from the stem it reaches the upper part, that is the leaf, where you can see the stoma. Through the stoma the water is evaporated or transpired into the atmosphere. Again during rainfall the water comes back into the soil and again it's taken up. So it is a cyclic process. Now further coming to the last part of the chapter, transpiration is a necessary evil. So even though it is necessary, but it's a necessary evil, cannot be avoided. For stomatal transpiration to occur, stoma must remain open during daytime. So if stoma is open, only then transpiration will take place, or vice versa. When stomata are open, then only gas exchange needed for respiration photons will take place. So with stoma closed, the respiration of photons will not take place. So it is necessary that stomata should remain open, specifically during the daytime. If stomatal transpiration stops, it will affect the productivity of the plant through the loss of photosynthetic and respiratory activity. That is, if the stomata is closed or stops, then the transpiration, that is, it directly affects, thereby also affecting the photosynthesis and the respiratory activity as well. That is, hence the productivity stomata must remain open. So to carry out this process like photosynthesis, respiration, it is necessary that stoma to be open. Consequently, transpiration cannot be avoided. So then transpiration will definitely occur as stoma is open. That is why Curtis in 1926 regarded transpiration as a necessary evil. So here added another image showing roots take up water from the soil, how the movement of water takes place underneath the ground. Then second step, water is drawn up the stem through this region, as you can see how through arrow it is shown very clearly. And then third part, water evaporates from the leaves. So side by side you can see this water passing through the stem from the root, then from the stem further into the leaf, and thereby transpiration taking place. So I hope students, the entire concept from the first part of the video, second and third, today's part is very clear. In today's part, as we learned about the transpiration, the types, stomata, structure, etc. Okay, so thank you all of you. Hope the concept is clear.